Hi everyone, welcome to the online version of my Evolution 2022 talk on using developmental rules to bridge the gap between micro and macroevolution in primate teeth that I've been developing in my current postdoc at Virginia Tech. Classically, we have classified evolutionary phenomena in two categories. One is the microevolutionary scale, where we evaluate changes within species at a restricted time scale and usually involves changes that are reduced in magnitude. On the other side, we have macroevolution, which involves the analysis of multiple species at larger time scales and involves changes of larger magnitudes. Two classical examples of these are the microevolutionary change of Darwin finches' beaks as a response to drought events and the macroevolutionary trend towards the increasing brain size in the lineage leading to humans. One of the greatest questions that students of macroevolution have considered is to what degree can we extrapolate microevolutionary processes to macroevolutionary contexts? Or in other words, if is macroevolution just microevolution plus time? If there is a discontinuity between these levels of analysis, then our ability to make inferences at large scales is compromised. As an historical science, macroevolution cannot directly test predictions about the fundamental processes of evolution, such as selection and drift, which are the realm of microevolutionary studies. However, if that bridge is closed, the connection between these two levels, then our ability to narrow into biological causes at large scales can be severely reduced. Then, what is the answer to this question? If I can summarize honestly the consensus of the field on this question, it would go something like this. Is macroevolution just microevolution plus time? Yes. Well, sort of. But in a practical sense, not really. But maybe? So what do I mean by that? So yes, because all that is happening at both scales is sorting of alleles through generations by means of natural selection and genetic drift. There is no additional phenomena, no magic going on that is necessary to explain the dynamic that happens at both time scales. But in a practical sense, when we apply microevolutionary models to macroevolutionary data, then we start to see some conflict. This graph shows how most macroevolutionary change is actually slower than the rates that would be expected under drift, suggesting an absence of directional selection in shaping large-scale trends, which is obviously nonsensical. However, some studies have shown that some macroevolutionary data fit some of the predictions of microevolutionary models. For example, if it has been shown in multiple groups that the rates of evolution of individual traits are proportional to the intraspecific variation on each one of those traits, something that is predicted under some microevolutionary models. This results in these paradoxes where some features of macroevolution seems to confront to microevolutionary predictions while others do not. To try to solve this conflict, we propose here a reframing of this issue. What if the issue is not arising due to inadequacy of the models to the data, but instead what if it relates to how we are quantifying biological information themselves? So what do I mean by that? For example, if we measure phenotypes with what I call naive rulers, so quantification methods that make no relation, no explicit assumption about biological processes, then we can get very complicated morphic spaces with a lot of discontinuities and dimensionality issues and etc. However, if we manage to derive a biologically informed quantification process that takes into account complex developmental rules, for example, maybe we can construct simplified morphospace that can lead to a better match between quantification and model. To test this possibility, we investigated the evolution of primate lower molars. So primate molars have some very interesting features there that are perfectly conducive to this kind of analysis. First, Primate molars are widely available in museum collections for both extant and fossil species, making this a perfect model to evaluate phenotypes on a deep time scale. In our case, we compile a data set from some large primary sources and supplemented those with additional sources and new data provided by multiple individuals, all evaluated under the context of a new comprehensive phylogeny, which contains both fossil and living taxa. So all this information was summarized and curated by two awesome undergrads, resulting in one of the largest data sets for a complex phenotype available to date. Second, and more importantly, there is a very well described and corroborated model for the development of lower molars called the inhibitory cascade model. This model dictates some rules that mammalian teeth phenotypes tend to follow. Specifically, this model says that the relative size or area of individual molar teeth will progressively increase or progressively decrease along the molar row due to the balance between inhibition and activation factors. So this delineates a natural morphospace defined by the relative sizes of the second and third molars in relationship to the first molar, making this a perfect subject for our kind of investigation. In practice, what we do is to measure individual teeth sizes, take the ratios between the third and the second teeth against the first one, and construct a morphospace to investigate phenotypic evolution. So here's an example of multiple mammalian species plotted on a classical graph for the inhibitory cascade model. And here is our data, and by just quantifying teeth in this case, voila, we have a biologically informed morphospace. So what we need now is to construct models that allow us to test if microevolutionary models can be actually fit to this kind of data. To evaluate this, we use the multivariate extension of the OU model under the framework of the mixed Gaussian phylogenetic models. The general form of these models is the one detailed in this differential equation, which describes the behavior of the phenotypes through time. This equation is divided in a deterministic portion and a stochastic portion. On the deterministic part, we have two main parameters, which are the optima theta and the matrix H, which controls the pull towards this optima. On the stochastic portion, we have sigma, which is the matrix that defines the stochastic rate of change, sometimes called the rate matrix. If H in this model is a matrix of zeros, then this model is a pure Brownian, multivariate Brownian motion model, just to show that this equation accommodates both models, most common models of phenotypic evolution, both OU models and Brownian motion models. Under a strict microevolutionary interpretation, these parts, the stochastic part and the deterministic parts, can be interpreted as a stabilizing selection portion and a genetic drift portion, with the deterministic parameters describing elements of the adaptive landscape, such as the optima and the rate of attraction towards the optima. And on the stochastic portion, we have the sigma, which now can be viewed as the rate of genetic drift. If we follow this microevolutionary interpretation, sigma is then no longer a free parameter, because the rate of drift will be proportional to the amount of genetic variation, here represented by G on these same traits. In other words, under a microevolutionary interpretation, sigma will be proportional to G. We then can use this framework to construct microevolutionary models, here in orange, to make this explicit assumption about how the rates of evolution will behave, and macroevolutionary models which make no such assumptions. We also fitted a mixed model that allow for multi-regime models to be proposed for the dataset. So all these models were then fit under a maximum likelihood to our data. Lastly, We tested this model not only for the inhibitory cascade variables, but also for naive morphospace quantifications, such as traits, linear distances between extreme points of teeth, and areas to evaluate the impact of morphospace choice on our conclusions. Okay, now let's go for some results. For both naive quantifications, we find evidence of a multi-regime macroevolutionary model, which allows parameter heterogeneity along the tree. For these models, our diversity was divided in 3 regimes, one ancestral one that also included tarsiids, and one for lemuriforms, and a third one for similiforms, which are monkeys and primates. For the linear distance models, the best model was a multi-OU model with each regime having their unique parameters. And for areas, the best model was a multi-Brownian motion model with each regime having its own rate parameters, for example. However, when we evaluate the inhibitory cascade model variables, the preferred model was the microevolutionary OU model, exactly like we predicted, showing that ontogenetically informed spaces can provide a bridge to connect micro to macroevolution. With this connection now made, we can use now microevolutionary theory to map phenomena at large scales. For example, we can both quantify the rates of genetic drift over millions of years and also visualize the adaptive landscape of teeth evolution in primates. Looking at these results, we can then conclude that the developmental processes, the inhibitory cascade model processes, seems to impose a corridor-like adaptive landscapes where primate species seems to be moving almost neutrally within it, producing all the observed variation in the fossil record and on extant species. In conclusion, it seems like the use of the developmental rules can allow a better match between micro to macro evolution, at least in the present case. The use of these models then allow us to dissect our macroevolutionary patterns in terms of their fundamental processes, such as adaptation and drift. And more importantly, this kind of reframing of the issue opens the door for the incorporation of more biological assumptions into our macroevolutionary studies. I would like to finish by acknowledging NSF and Virginia Tech, and more importantly, Dr. Ueda for giving me the opportunity to develop this project. If you have any questions, please reach out to me over email or on Twitter. I would also like to take the opportunity to say that I'll be moving shortly to Oklahoma State, and if you find any of these topics interesting, and if you're looking for a PhD program or a postdoc job, please feel free to contact me. Thank you for your attention and have a wonderful day.